call and ask questions, but I can't hear them, so we need to have them show up in the chat box. After this section, I'll check the chat box again and answer anything that's there. Okay, before launching into the next topic, I want to first highlight some of the advantages of constructing a formal experimental design, which is what we're going to talk about now. And this is where you actually write out your hypotheses and their implications, and then follow a series of defined protocols that we'll be talking about in your experimental work. First of all, this prevents unintentional bias, and that will allow you to be more confident that the results of a small study of a few objects or simulated objects can be generalized beyond that to actual treatment decisions. You're also more likely to have accurate results. That's of course important. And the risk of self-deception is avoided or reduced by attention to design choices. And we'll look at a few examples of when that happened. In addition, some elements of a formal experimental design are required for meaningful statistical analysis. And in medical and agricultural research, this approach has brought about many great developments, and I think the same is likely to be true in conservation and preservation research. There are three divisions where decisions have to be made within the design of experiments, and therefore these mark the major aspects or steps of an experimental design: your object protocols, measurement protocols, and treatment protocols. So we'll be discussing each of these in turn. For object protocols, the major decisions to be made include whether to use real objects or facsimiles, and how many replicates to test. As an example, for tests to identify the materials or technology used in making a ceramic pot, we need to decide how to conduct testing or sampling protocols for the pot itself. If we can test only a few objects out of a larger group available, if these are randomly selected, at least we can then generalize with some confidence to the collection itself. So we know that our results will be something that is relatable to the entire collection. If we've randomly selected test objects from several sites or collections, we can then generalize even further to this particular type of ceramic. Or we may have a ceramic tradition with many similar sherds, and these are still real objects versus typical experiments using facsimiles, things such as metal coupons that were constructed for a test. If we can't sample real objects, or if we prefer to begin a testing program first using facsimiles before sampling real sherds, then we can make ceramic test tiles that simulate the composition and structure of the sherds. For studies characterizing a material or technology, of course, we need the real objects. But what about tests of treatments? For example, I once observed a pot that had been damaged by multiple black spots along the surface. Someone had spot tested a potential surface cleaning method on a real object, which happened to be very porous and filled with carbonized material within the microstructure. The applied solvent in the test drew the carbon up through the network of pores very quickly and deposited it on the surface. There were a lot of sherds stored along with the pot, so clearly those would have been much better for the initial testing, and thin sections made from one of the sherds actually would have revealed this very porous carbonaceous interior, and we would have seen this pore network that would have brought the carbon up. So not starting testing on simulated objects or similar sherds can have a very disastrous effect. when we then go and put something on in the actual whole object. There are a lot of examples in the literature of testing something like a new cleaning method on real objects, not always with mention of whether or not prior tests were done on simulated objects. Even if the new method doesn't cause damage immediately, it's often unknown what the long-term effects might be. And one cautionary tale was reported in a letter to the editor in Studies in Conservation back in 1991 concerning a treatment for rock art sites that was published in the journal more than a decade earlier. There was an artificial desert varnish that was chemically induced over the rock art of many ancient petroglyph sites in the American Southwest desert as a measure that was intended to protect the engravings from damage. But over time, it turned out that this artificial varnish darkened and had actually begun to obscure some of the petroglyphs, and it also began to show some sharp lines of demarcation with areas of natural desert varnish in the surrounding rock. And so it's becoming difficult to view the petroglyphs in the form that they'd originally been created. But removal of the now discolored artificial desert varnish was not possible because it would have been too damaging to the stone and the petroglyphs. So in this case, before any treatment was applied to real rock art sites, it would have been useful to apply the treatment to simulated rock art and then do artificial aging to see how well the treatment held up over time. Typical experimentation in conservation and preservation involves experiments using things like paper strips, metal coupons, or the like, rather than on real objects. An example would be testing how well metal coatings hold up under long-term exposure, or how specific metals react to pollutants in the environment. Subjecting real objects to the test conditions would make no sense because it's too risky, requiring cleaning and polishing and damage if we're wrong about the potential materials or conditions. So we use simulated coupons, for example, of the appropriate metal type, as might be found with collection artifacts. We need to clearly define those simulated objects to be as close as possible to real objects, such as deciding which metals to test, whether to use fine or sterling silver, etc. One mistake that scientists from other fields sometimes make in testing treatments or coatings for conservation use is to use substrates not typically found in ancient or historic collections. But how much replication to do is also a crucial issue here. The issues with replicates include how many do we need and how do we choose them. The main purpose of replicates is to identify random variation in objects, measurements, and treatments so we can, first of all, estimate variability, and second, estimate actual treatment effects more accurately. We can't always find a way to have replicates with real objects, but there's really generally no excuse for simulated objects not to have replication. We need some replication to do statistical analysis and to avoid random odd results or experimental blunder. And remember back when I mentioned that if something can go wrong, it probably will. So we need replicates. But if we have too many replicates, then we can needlessly complicate things or add on too much time and effort without gaining appreciably more in terms of results. One example is with Oddy tests. A lot of people don't do any replication at all, but sometimes we do see variation within replicates being tested with the same materials, or even variation in the appearance of the controls, making interpretations difficult or even potentially inaccurate. Sometimes something goes drastically wrong, like you forgot to put water in one vessel, or put too little in, or the vessel wasn't well sealed. So having at least 2 replicates can help you identify where something may need further follow-up testing. A paper on your reference list by a man named Appelman back in 1990 noted that too much testing in the coatings industry used no replicates, and so was not actually reliable. And he pointed out a number of cases where everything was actually quite wrong. So I'm not sure this problem has really improved since then, and you can see it's not just a conservation and preservation problem. But how many replicates are needed depends on the variability that you expect. If you have no idea what that might be, then starting with 2 or 3 replicates is pretty reasonable. If the variability is expected to be quite large, then this should be increased. However, experiments involving human beings or other highly complicated and variable biological organisms often consider that 8 to 25 replicates is generally considered sufficient. So it's wasting time and effort to go too wild with replication beyond what will actually give you new information. And looking at what constitutes a replicate, or how we define or choose them, we need to think carefully about the purpose of replication. In an artificial aging study of an adhesive applied to paper, for example, we could just apply the adhesive to one sheet of paper and then cut it into 3 pieces, and so we have 3 replicates. But if we think there may be variation in the paper, and that could be especially true if we're using a historic paper, or if we think there might be variation in our application process, maybe it might be a bit thinner in some areas than others, for example, then using 3 separate sheets of paper with 3 separate applications of adhesive will better catch any potential variation. And if we had to prepare the adhesive mix in-house, or have some reason to think that there might be variations in purchased batches, then we may even want to use 3 separate batches of adhesive. For testing a treatment to be applied to natural history skins, for example, we know to expect some variation because we're working with a complex biological specimen that's also been treated in ways that might involve highly variable preparation methods. On the other hand, even simulated objects, skins that we acquired and prepared ourselves, are difficult to acquire and time-consuming to prepare, so we do want to be judicious in replication. We could use only one skin and then apply 3 different treatments, each with replication, to different patches. And later, when we talk about single object studies, we'll look at how that can be done in a statistically valid way. But if we expect a lot of variation in the substrate, we can get 2 separately prepared skins from 2 animals, and then we can apply the same number of replicates of each of the 3 treatments, but have each applied to a different skin. So it really depends on where we expect variability and what's reasonable and doable, but still able to catch that expected variation. Things to consider with measurement protocols include deciding on variable types. Those are going to be quantitative data, presence-absence data, numbers that represent categories, numbers that represent real quantities or amounts, etc. Other issues to consider are how many repeated readings are needed, and we'll look at each of these in a moment individually. How many repeated measurements, as well as various solutions to help us try to avoid bias. The purpose of repeated readings is to increase accuracy by averaging out measurement error, such as you might get with an instrument noise or reading errors. So we're going to here want to average the data. And protocols often exist for the particular instrument that you're using. With repeated measurements, here we're taking measurements at different spots or at different times. And the purpose is to detect any spatial or temporal variation or change occurring in the sample. So spatial variation might be edge effects. Temporal variation, the most common thing we're looking at is before and after treatment, or before and after accelerated aging. So these are not averaged. Now, how many do we do? It depends on the importance and the expected significance of the time and space differences and the likelihood of there being a measurable difference, or at least one that matters. So these should be limited to what is really likely to be important. And so you're going to want to have clear rationale for any repeated measurements that you're taking. You want to have a number that's both reasonable and productive. For example, if you have no reason to think that there's any spatial difference, then you still might want to measure a spot on the interior and a spot on the edge of each specimen, but you're not going to want to measure 6 or 10 different locations. That just wouldn't make any sense. It's a waste of your time. And if your main question is whether there's a significant difference in the treated area before and after accelerated aging, then it doesn't really make sense to also add in measurements that involve removing the specimens from the aging chambers at a lot of different intervals along the way. You might want to do that if your research question is, at what point does change occur? But if you only want to know, did change occur after aging, then you're wasting a lot of time to take a lot of interim measurements. Avoidance of biased experiments is a crucial consideration, because otherwise you can expect that you may be getting a wrong answer because of poor research design. One classic example is that of N-rays. In 1903, a very distinguished French physicist announced a discovery that he'd made of a new form of radiation. Objects placed in an X-ray beam directed through a prism would then emit light that could be seen in a phosphorescent detector, and he named these N-rays. While many researchers claimed to have replicated the experiment, an equal number just could never replicate it. They could never see the N-rays. Finally, someone visited the original physicist's lab, and without the French physicist's knowledge, removed the prism during the experiment and replaced it with inert wood. So no N-rays should have been produced, but the physicist still claimed to be able to see the N-rays. It turned out that, in fact, they don't exist. So once this whole chain of events was reported, no one ever saw them again. That was the end of N-rays. And we'll be looking at what the problems were here, and lack of blinding and randomization led to this result. Sometimes a person may feel their career, for example, depends on a specific technique being the best, or institutions may have spent millions of dollars on an object, and now it's been questioned, and so they want you to authenticate it. There's a lot of pressure there. So these things can bias us, and without us trying to, it can lead us to see things that we want to see and to not see things that we prefer not to be there. So it's best just to accept that this can happen, and then choose techniques to avoid bias so we don't have to worry about it. Standard techniques for doing this include randomization of measurement order or measurement place, and blind analysis, not knowing which objects received which treatments while you're taking your measurements. And so, of course, when we're doing this, we need to make sure we have some way of adding numbers on here or labels, so we have a unique identification code that we can track each particular object through the blinded or randomized measurement process. E. Bright Wilson wrote an excellent book on scientific method that's on your list of references, and he gives an example of the need to randomize measurement order. This was a case in an industrial lab where experiments were done to check the effect of length of time of pressing of plastic parts in the mold on the strength of the finished plastic parts. Hot plastic was first pressed for 10 seconds and removed. The next batch was then pressed for 20 seconds, etc. And the results showed a strong dependence of length of time pressed with strength of the part. But the supervisor rejected the results because pressing times had not been randomized. The lab then redid the experiments with randomization. It was clear now that the dependence didn't actually exist. It appeared that because the mold got hotter and hotter during the experiments, it was actually heat of the mold, not length of time being pressed, that was related to strength. Another example Wilson gives us that in one lab carrying out a set of chemical analyses, standard practice was to run pairs of duplicate analyses together, and agreement was good. But another lab found a lot of discrepancy in their results. They couldn't duplicate the results. And it turns out that a zinc reductor, through which all the samples ran, gradually lost effectiveness over the day because of the presence of certain other elements in the sample. The effect from one sample to the next was small. If they had randomized the order so all duplicates were not done side by side, they would have seen major errors. Another common example following the similar idea that we see in conservation is that a chromometer can often drift gradually. So even if we calibrated it at the outset, if all the replicates of one treatment or coding, etc., were measured in order, the results might be different than if all of the treatments and codings, etc., were randomized for the measurement order. Blinding refers to taking measurements without knowing which sample received which treatment. Now, this can sometimes be difficult or impossible to do with cultural heritage objects, but it's especially important to try to do for subjective measurements or assessments. An example is the use of the placebo in medicine. The physician who's assessing how well the patient's responding doesn't know whether the patient received the proposed new treatment or a standard treatment or even an inert placebo. An example in conservation is the grading of Oddy test coupons. And these are assessed as similar enough to the controls to be considered passed for use as a storage or exhibit material, a little worse than the controls but good enough for temporary short-term use, or so much worse than the controls that they should be considered failed for any use near metal artifacts. I found that if I know which two coupons are replicates that were housed with the same test material, then my mind expects that they should perform the same. And if one of them is borderline between a pass and a temporary, or between a temporary and a fail, and I know which way I graded the other replicate, I tend to see it the same way. But if instead there are stamped numbers on the back or some other way to code the coupons for grading without knowing which test materials each have been housed with and which ones were replicates of each other, I often graded things differently. So the same principle applies to grading the performance of coatings or any other treatments. This issue is less important maybe for automated measurement recording, but it's crucial for subjective assessments. Important things to consider regarding treatment protocols include controls and randomization. Controls receive either no treatment or a standard comparative treatment. Their main purpose is to be certain that the effects that we see are actually due to the treatment and not to chance or to some uncontrolled factor. But controls are not always needed. We need to think about the purpose of the control. For example, if the experiment is intended to compare one coding to another, then we might not need a third sort of control. Randomization of objects to treatments is important for a number of reasons. First, statistical tests assume treatments were assigned by random process. It's an underlying assumption of many statistical tests. Well, yes, of course, you can still run a statistical package using your data if you didn't randomize specimens to treatments. Alarms aren't going to come on and the program won't freeze up. There won't be red flashing lights showing up on your computer. But your results will not necessarily have validity or be reproducible. And you'll find that if you try to work with a professional statistician, he or she will likely refuse to do the analysis. Second, randomization prevents bias in selection of which objects get which treatment. And we'll look in a moment at cases where that has been a problem. And third, it distributes uncontrolled factors among all the treatment groups, preventing bias that will accidentally make one treatment effect appear to be different from another when it's actually not. One cautionary tale that E. Bright Wilson discussed in his book was a study of whether a program of offering milk to children during the school day would have an effect on their height and weight over a 3-month period. This was a large-scale experiment. They had 10,000 children selected as test subjects who would get milk, and 10,000 as controls who would not get milk. So obviously there's no problem with the amount of replication here. There was initial randomization of children to treatment, milk or not. However, fatal flaw in the experiment was that teachers were allowed to change that selection if they felt their class appeared to be unbalanced in the number selected for each group. And it turned out that the natural human sympathy of teachers created a very biased sample. Children that were already taller and heavier than their peers were assigned by teachers to the control group because, after all, they didn't really need more milk, did they? And those children who appeared to be undernourished, teachers felt sorry for them and they assigned them to receive milk. So in the end, the biases prevented any clear result. So this huge and costly experiment had no reliable results to show. Unlike popular conception, randomization is not haphazard picking out from a group. Instead, the term randomization refers specifically to use of a variety of methods that have known statistical odds that can be calculated. Haphazard selection is not random because there can still be an underlying bias in the order in which things are selected or assigned to a treatment group. Selecting laboratory mice to be placed in one treatment group or another, for example, is always done by a proper randomization method because it was long ago noticed that various types of biases otherwise emerge. For example, if we say that the first 10 mice we happen to pull out from the cage are going to go into treatment group A, and then the second 10 that we pull out will go into treatment group B, our experiment will be worthless. We can't trust that any significant difference in outcome seen between the two experiments, between the two treatments, has anything to do with the treatment itself. And why? Well, because the healthiest, strongest mice may run away from our hand, leaving the sickliest, tired, old ones to be grabbed first. So if the first treatment appears to result in more sickly or dead mice, well, it's just that more of them may have started out with health problems to begin with. Randomization of objects to treatments requires a bit more time and effort, but even in conservation and preservation, where our subjects don't run away or try to bite us, not randomizing can affect the outcome. For myself, if I'm not randomizing, I found myself subtly choosing the order of specimens to group based on something like what's easiest and fastest to set up. There are several methods for accomplishing randomization by methods of known statistical odds. And the easiest one often depends on how many treatment groups you have. Flipping a coin is one method, and rolling a die, or you can use multiple dice and combining the numbers if you have more than 6 groups. A random number table can also be used. So here you decide ahead of time where in the sheet you're going to start and what direction you're going to move in. Depending on the number of groups, you can use single digits or double or even triple digits. If you land on a number you've already used, then you can move on until you land on a new number. And in the same way, you can use a computerized random number generator. These are actually called pseudo-random number generators because they satisfy statistical tests for randomness but are still produced using a definite mathematical procedure. But be careful about the program you use. Do some research to see if it's accepted by statisticians as a pseudo-random number generator. A colleague of mine once used a website that was casually found, which claimed to generate random numbers, but once she carried out the assignment of specimens to treatments using it, she noticed there actually was a clear patterning, so it wasn't random at all. But don't just pick up any old website. Major statistical packages like SPSS, developed by teams of statisticians, for example, have reliable pseudo-random number generators. You can also draw numbers or treatment groups out of a jar, and this can be an easy way to randomize assignments for a relatively small set of specimens and treatment groups. However, this will only randomize if the pieces of paper you use are very well mixed and if you can't read any of them while you're drawing. So this is the same basic principle as a lottery. There are different ways in which to construct a randomization procedure. The first is with object fixed. You've decided which object to assign and then select the treatment group number using a randomization procedure. So in this case, for example, you might be drawing A's and B's out of a jar. First to assign a treatment group to sample 1, then to sample 2, etc. The second is with treatment fixed. You start with a particular treatment group and then fill it by assigning objects through the randomization procedure. So here, for example, we would start with treatment A and then randomly draw numbers until we had 3 samples that got assigned to that group. You can develop a stratified random sampling strategy, restricting in some way the population that you're going to be selecting from. For example, if you're studying the pigment use of a 19th century artist, you can't sample the painting in random locations, but you can at least report what was done and how it might affect interpretations. So if you never find a particular pigment, it might be because certain color areas could never be sampled and you need to report that. But stratified random sampling might be possible along the edge, along the frame, under the frame, from each color area, or maybe just where there are existing cracks in each color. But if you can't sample some colors, you need to take account of that in reporting and interpretation. I've seen papers reporting on the pigment use of a particular artist that were organized with tables organized by color, as if this were the full palette of the artist. But then looking at the images in the publication, you can see that there are some pigment colors that are completely absent in the reported chart, which is sort of puzzling. And a final example of unforeseen problems that can occur and affect results if randomization isn't done is from a paper project discussed in one of my favorite books, The Cartoon Guide to Statistics. I highly recommend it. Here they're talking about an experiment that was testing gas mileage for 2 different types of gas formulations using 2 taxi fleets. So one fleet was assigned to drive on Tuesday using one gas formulation, and the other on Wednesday using the other gas formulation. And a significant difference in mileage was found, but it turned out that the difference had absolutely nothing to do with the gas formulation. What happened was that on Tuesday it rained very heavily, so that taxi fleet had to drive a lot more cautiously, and it turned out that accounted for all the difference in mileage. Because studies show that accelerating more slowly away from green lights and then stopping more gradually for red lights can cut fuel consumption down by as much as 35%. And this is also a driving behavior that drivers are more likely to follow in a heavy rain with slick roads. So it would have been better to randomize the gas formulations to individual cars or cars to test days so that there would be fewer uncontrolled variables. I invite you to go through the randomization practice exercises that are posted on the NCPT website. These are intended to help you go through and try out various methods of accomplishing randomization, and it includes a random number table as well. So we'll try taking a few questions again here. If you have any